So this would be activity 5, media preparation. So this would be the first topic for the midterms. Now, what is the reason why we do media preparation in bacteriology? Now, media preparation class is usually done to give a better result. Now, as you all know, direct methods such as microscopy and nucleic acid detection would provide early information about bacteria and fungi in infection. However, microorganism growth is usually required for confirmation, identification, and characterization. Ganito kasi yan, class. If you still remember in your activity 4, nag-perform tayo ng gram stain on a throat swab. Now, upon viewing the microscope, you were able to see different types of bacteria. Gram positive cocci in chains. Some were in clusters, some were in pairs, etc. Now, the thing here is that whenever doctors or whenever we medical technologies would report gram stain result, there is an equivalent, meron niyang equivalent na corresponding bacteria. Let's say you reported in your gram stain gram-positive cocci in chains. This could be a identification or an identifier for the bacteria Streptococcus pyogenes. Kapag naman ang gram stain mo, if your gram stain naman shows gram-positive cocci in clusters, this is a presumptive identification of the bacteria S. aureus. So it basically gives class a screening identification for what possible bacteria that is. But again, you need to confirm this. By confirming this class, the doctor would have a better diagnosis. And for you to confirm that, you need to do media preparation or what we call yung culture. Now, what are the principles of bacterial cultivation? Now, bacterial cultivation has three main purpose class: to isolate bacteria from clinical specimens. Now, we need to isolate the target bacteria from the clinical specimen class because in some cases, yung mga sample na pinapadala has more than one type of bacteria. So, we want to look for the pathogenic. We need to look for the pathogenic bacteria. The second one is to determine which bacteria are most likely causing infection and which are contaminants or normal flora. Now, sa body natin kasi class, we have what we call normal flora. Ano ba tong normal flora na to? Now, if you're still unaware, ang normal flora natin are bacteria or microorganisms that normally reside in our body. The most common normal flora na ma-encounter nyo based on your lecture is si Staphylococcus aureus. Nakikita siya sa skin and even other body parts. Now, we need to do this culture to isolate or to differentiate if it is a normal floral bacteria or a pathogenic bacteria. Why? To give you an example, class, let's say ikaw, yung klase ng patient that would continuously take, mahilig ka uminom ng or mag-take ng Listerine or other na oral mouthwash. Now, what happens if you take Listerine too much? There's a possibility that it would kill the normal flora in your mouth. And when that happens, the normal flora would be replaced by pathogenic bacteria. Kaya nga in some cases, kapag take ng mouthwash, even though nagiging normal na yung bad breath sa'yo, kakatake mo ng mouthwash. The reason for that is because the normal flora has already been replaced by pathogenic bacteria. Now, the third reason is to obtain sufficient growth for confirmation, identification, and susceptibility testing. So sa mga cases class katulad ng mga sample na CSF, yung mga sample that usually have very low amounts of bacteria, we need to try to propagate them, and to do that we have to culture. Pag napadami kasi natin sila, we can do more tests allowing for better diagnosis. Now, what are the nutritional requirements? Now, your bacteria class in culture requires different nutrients such as gases, water, ions, vitamins, minerals, nitrogen sources such as peptones, casein, meat, or plant infusion, carbon, and energy. Now, carbon class is usually supplied through carbohydrates. So, ang carbon source na ginagamit ng bacteria are usually from the carbohydrates, yung mga sugar, yung mga lactose, sucrose, fructose, as well as protein. Now, successful cultivation depends on providing essential nutrients and an appropriate environment for growth. Now, what are the general concepts of a culture media? In the laboratory, nutrients are incorporated into a liquid or solid media. If the media meets bacterial requirements, cells will multiply to visible level. So there are two types of media class, a solid media and a liquid media. So later i-explain ko kung ano yung mga yan. Now in liquid broth, growth produces turbidity that can be seen with the naked eye. For accurate interpretation, the media must be placed in the right environmental condition. Bacteria with demanding requirements are considered fastidious, while those with simpler needs are non-fastidious. Pag sinabi mong fastidious class, ito yung mga bacteria that would require special nutrients and special condition for them to grow. The most common example of this is yung Haemophilus nyo. So si Haemophilus nyo is the most common example of a fastidious bacteria. Now, what are the phases of growth media? Now, growth media are available in two main phases, so the liquid and the solid media. Now, the liquid media is known as your broth. Please take note of that, broth ang tawag sa kanya, while the solid media are known as your agar. Now, in some cases, a biphasic media, both liquid and solid phases, such as certain blood culture methods, or a semi-solid medium, such as a thioglycolate broth with a small percentage of agar, may be used. So, ulitin ko class, your growth media are available into two types, liquid, known as your broth, and solid, known as the agar. But in some cases, class, they would be biphasic, meaning both liquid and solid, and in some cases, a semi-solid media, such as your thioglycolate, which only has a very small percentage of agar. Now, let's talk about the broth media. So, si broth media nyo class are dissolved in water, allowing bacterial to multiply in suspension. Now, growth is indicated by a visible change in the broth from clear to turbid. This turbidity results from light being scattered by bacterial cells. At least 10 raised to the 6 bacteria per milliliter are needed for the turbidity to be seen by the naked eye. And some broths include pH indicators such as phenol red, which can change color in response to bacterial metabolism. Now, sa broth media class, ang sign, whenever you would want to know if there is bacterial growth, the sign for bacterial growth in broth media or in liquid media is the presence of turbidity. Usually, class, ang mga broth media nyo, they are clear. So, if they become turbid, it signifies that there is bacterial growth. Now, aside from turbidity, there are also other signs where it would indicate there is bacterial growth. If you look at the surface of the broth, yung liquid media nyo, if there is a formation of a pellicle, yung pellicle nyo para siyang plug. Parang bacterial landmass. May landmass doon sa taas sa pinaka-top ng broth media nyo. That's a sign of bacterial growth. Also, if there is a presence of a ring, so dun sa edges ng surface, may parang growth, it could also mean bacterial growth. Then if there are sediments formed, bacterial growth din siya. If there are flocculents, yung mga malalaking buo-buo na colonies class in liquid media, and also your turbidity. So these are the different signs of bacterial growth in a liquid media. Sa surface, pellicle and a ring. Sa pinakababa, presence of a sediment. And in the broth itself, a flocculent growth or a turbid growth. Now, take note, class, that the location of growth in broth can also reveal oxygen requirements. Now, ang mga bacteria nyo, class, if it was already discussed in the lecture, your bacteria have oxygen requirements. Now, the thing you need to take note here is the so-called aerobes and anaerobes. Pag aerobes, they require oxygen for growth. Pag anaerobes, presence of oxygen would kill them. So, bawal may oxygen. Now, if your bacteria in broth medium grows at the bottom of the tube, so nagkaroon ng sediment, ang growth lang ng bacteria is nasa baba lang, this is a sign that your bacteria is a strict anaerobe. Meaning, class, pag na-expose sila sa oxygen, if they're exposed to oxygen, they would die. While if there is growth naman at the surface, nagkaroon ng pellicle, nagkaroon ng ring, there's a pellicle, there's a ring, it means that your bacteria is an aerobe. They require oxygen. While microaerophiles naman, class, pag sinabi mong microaerophiles, these are bacteria that will require a small percentage of oxygen. Usually, mga 5 to 10% lang yan ng oxygen, class. The growth would just be below the surface where oxygen is lower than atmospheric level. So, kung ang growth niya, class, nandito lang, just below the surface, there's a possibility that it is a microaerophile. Then, if it is facultative anaerobe and aerotolerant, it would grow throughout the broth. Now, pag sinabi mong facultative, if you say it's a facultative, it may grow with or without oxygen. So sa case na to class, facultative anaerobe siya, di ba? Ang anaerobes nyo, they are not allowed to be exposed to oxygen. But if they are facultative anaerobes, okay lang. They can still grow even in the presence of oxygen. So growth would be spread throughout the broth. So kung turbid siya, may mga floccules, possible na facultative anaerobe. Now, let's go to the solid media nyo. Now, your solid media class consists of nutrients plus a solidifying agent, mostly commonly known as your agar. Now, agar melts at around 95 degrees centigrade and solidifies below 50 degrees centigrade, making it ideal for lab use. Now, types of solid agar preparation class, there are 3 types. Yung plate, yung slants, and yung dip. Now, pag plates class, these are wherein you would use a petri dish. You would pour the media on a petri dish. For slants naman class, tubes are placed in a slanted agar surface, useful for storage and maintenance culture. Then, ang mga agar dip. Now, another name for agar dip class is yung tinatawag nating agar butts. Tubes filled to create a deep agar column used for studying oxygen requirement or motility. Now, solid media supports the growth of visible colonies, each theoretically arising from a single bacterial cell and therefore representing a pure culture. Now, if you take a look at this picture class, ito yung mga plates nyo. So it's media, basically media in a petri dish. Now, ito naman yung mga slant niya. So, ito nakikita niya, this is the slant. Ito naman yung mga butt or dip nyo. So, in some cases, class, meron tayong tinatawag na butt slant na media. This is a combination of the two. So, meron siyang butt and meron siyang slant. So, this is an example of butt slant. Ang mga slant class, kung ito ang tube nyo, they tend to have a very, very long slant with a very small butt. So, ganyan ang slant nyo. But if it's a butt class, a butt or a dip, maikli lang yung slant, puro dip na. Ayan. But if it's a butt slant, pag butt slant na combination, mahabang slant siya, malaki din yung dip or yung butt. So that's the different types of solid media. The plates, the slants, the dips, or the butt, and the butt slant. Now let's talk about the importance of colonies. Now when your bacteria class grows, they tend to grow combined, or they tend to attach to one another, they tend to group up, and they would visibly form what we call colonies. So ito yung mga colonies yan. So these are groups of bacteria class attached together. Now with appropriate incubation, individual colonies can be selected for further testing. A pure colony contains genetically identical bacteria, which allows accurate identification and characterization of the organism. The ability to select colonies is one of the most important steps in bacteriology, since only pure culture can be reliably studied. So if you still remember sa lecture nyo, your bacteria would usually multiply by the process known as your binary fission. Si fission nyo is the opposite of fusion, di ba? Si fusion to combine, si fission is to separate. So if you have a single bacteria, through binary fission, it would double. Magdalawa siya. Then, they would double again, and so on. Basically, it's like an exponent class. Diba? Sa exponent, they tend to double. So, your bacteria, if you remember if naturo na sa inyo sa lecture, yung stages of bacterial growth. So, one of those is the exponential stage where your bacteria continuously multiplies. So, this is from a single clone. From a single clone class, dadami siya ng dadami via binary fission. Now, you need to target this pure colony. Dapat isang klase lang ng bacteria. Now, what are the classifications of pure media? Now, there are 4 main groups of pure media class: the enrichment, the nutritive, selective, and differential. Now, many culture media can also serve more than one function, such as being both selective and differential. So, kung may 4 main groups class, enrichment, nutritive, selective, and differential, there is also a chance that your media can serve as 2 or more functions. So one media can be both selective and differential. Another one would be nutritive and differential. Another one would be enrichment and selective. So pwede siyang 2 or more. Now let's talk about them one by one. Let's start with enrichment media. Now enrichment media class basically enhances the growth of a single or a particular pathogen. So they would only target a single group. Let's say ang gusto mo lang patubuin is Staphylococcus aureus. You use an enrichment media targeting S. aureus. It would only allow the growth, or it would only support the growth of S. aureus. Basically, it provides a growth advantage over others. A good example of this is yung buffered charcoal yeast extract agar nyo or yung BCYE, which contains L-cysteine and other nutrients essential for the growth of Legionella pneumophila. This is the causative agent of Legionnaire's disease. Now, si Legionnaire's disease nyo class, this is a pulmonary disease. And commonly, ang Legionella nyo is usually found in aircon cooling towers. Now, your enrichment media usually exists in the form of specialized broth, usually used to recover organism in very low numbers. These are particularly useful when initial inoculation on solid media does not yield visible colonies. So, ang example ng mga to class ay yung thioglycolate broth nyo, which supports anaerobes, limb broth, which is a Todd-Hewitt broth with colistin and nalidixic acid, used for the selective enrichment of group B Streptococcus. And there's also the GN broth or the gram-negative broth, which enhances the recovery of enteric gram-negative bacteria. Let's go to the second group, the nutritive. So this supports most bacteria class. So ito yung pinaka-routine. These are the routine media that are usually used in the laboratory. They grow the majority of your bacteria. They do not favor one type of organism over another and are widely used. Example is your tryptic soy agar, nutrient agar, Saborude's dextrose agar. Now, these media are the workhorses of bacteriology, allowing a broad spectrum of bacteria and fungi to grow. They are often used as a baseline to compare growth on selective or differential media. Then we have your selective media. Now, si selective media naman, class, they inhibit the growth of certain bacteria while allowing others to grow. So, nag-inhibit siya, class, at the same time, inaalaw niyang mag-grow yung target. Ang difference niya sa enrichment, binibigyan ng growth advantage, pero hindi siya nag-inhibit. Sa selective, it inhibits other organism. Now, it would contain different inhibitory agents such as yung dyes, bile salts, alcohols and acids, antibiotic. The most common example is your phenylethyl alcohol agar with 5% sheep blood. It inhibits the growth of aerobic and facultatively anaerobe gram-negative rods, permits the growth of gram-positive cocci. Now, selective media helps suppress normal microbiota or contaminants that would otherwise overgrow pathogens. However, limitations include such as that the inhibited organisms are not completely eliminated and they may still be present in small numbers. Improper incubation, such as dehydration of the medium, can weaken the selective effect, allowing contaminants to grow. And the last group is the differential media. So differential media contains ingredients that allow colonies of different bacterial species to be distinguished based on observable difference, such as a colony color, appearance, or biochemical activity. To simplify this class, the differential media nyo, it separates two groups of bacteria. So, kaya niya mag-differentiate between two groups of bacteria. The most common example is yung MacConkey agar nyo. So, it acts also as two types of media. Si MacConkey nyo is selective. It inhibits gram-positive organism. It's also differential because it separates gram-negative bacteria into lactose fermenters and non-lactose fermenters. Now, earlier class, sabi kanina, a source of carbon, di ba ang carbon nyo is required for bacterial growth, is usually coming from carbs and protein. Now, if your bacteria uses carbohydrate lactose, gumagamit siya ng lactose as a source of energy, it ferments lactose. The bacteria in MAC would be pink colonies. But if they are non-lactose fermenter, meaning hindi sila gumagamit ng lactose as their energy source, the colonies would appear colorless. Another example is the sheep blood agar. So this is a nutritive media. It supports the growth of most non-fastidious organism. It's a differential because it allows observation of hemolysis pattern, alpha, beta, gamma, which are useful in distinguishing bacterial species. Differential media are vital because they provide visual or biochemical clues about the organism's identity while still permitting growth. So later i-explain ko yung three types of hemolysis pattern plus sa blood agar, yung alpha, beta, and gamma. Now let's talk about the different media for routine bacteriology. Let's start with the most common one, the blood agar. One of the most widely used media for routine bacteriology, supports the growth of nearly all clinically significant bacteria except the most fastidious. It composed of a protein source, sodium chloride, agar, 5% sheep blood. Rabbit or horse blood may be substituted for specific organisms such as your Haemophilus. It enables the observation of hemolysis pattern caused by bacterial enzyme that lyse red blood cell. So si bacteria nyo class, when it grows, there's a possibility that it would secrete different enzymes. And in some cases, these enzymes would cause hemolysis. Kaya niya mag-breakdown ng RBC, producing hemolysis. Now, there are three patterns, beta, alpha, and gamma. Now, if it's a beta hemolysis, there is a complete clearing around the colony. Pag alpha, partial hemolysis with a greenish discoloration. And kapag gamma hemolysis, no effect on RBC. Now, your blood agar is important for colony morphology and hemolytic pattern, which is essential for differentiating species but should always be combined with gram stain and biochemical tests. Now, for you to better understand yung tatlong hemolysis pattern, ipapakita ko siya sa inyo. Let's start with alpha hemolysis. Now, a alpha hemolysis class is also known as your partial hemolysis. Now, it would produce a greenish or brownish discoloration around the colonies. It is partial or incomplete breakdown of RBC caused by bacterial hemolysis. The most common example of bacteria under this is your Streptococcus pneumoniae and your viridans group Streptococci. Only partial lysis would produce green halo from biliverdin production. So pag tiningnan nyo tong colony na to na tumubo, pagka-strict niya class, it produces a greenish discoloration, kulay green. So yan yung sign if it's alpha hemolysis. It shows a greenish discoloration. Let's go to the second one, yung beta hemolysis. So if it's beta hemolysis, it produces a clear, transparent zone surrounding the colonies. Complete breakdown of RBC. Most common example is yung group A strep nyo, yung S. pyogenes and S. aureus. Full lysis would produce clear halo. So if you take a look at this picture, ito yung colonies class. Surrounding the colonies is a clear zone, meaning nawala yung mga red blood cell dito sa part ng because there was total breakdown. And the last group is yung gamma hemolysis nyo. Basically, no hemolysis, no change. Red blood cells remain intact. Example of this is Enterococcus fecalis and some non-hemolytic Streptococci. So those are the three types of hemolysis pattern shown in your blood agar. Let's go to the second one, yung brain-heart infusion nyo. So this is a rich medium for both fastidious and non-fastidious. Can be both prepared as a agar or broth. Composed of animal tissue, peptones, phosphate buffer, and dextrose. Provides readily accessible energy for many bacteria, often used for culturing blood from patients. Suitable for antimicrobial susceptibility testing and establishing bacterial identification. So ito yung itsura ng BHI agar nyo. Now let's go to the third one, yung chocolate agar nyo. So chocolate agar is an enriched medium for fastidious organism that cannot grow on blood agar. Same as blood agar, but blood is added to molten agar lysing RBC. Ang preparation kasi class ng BAP. So here you would have your BAP. You would use nutrient agar. Then you add blood. Pagka-add ng blood, you would have your nutrient agar. Then you let it cool. Siguro mga 50 degrees centigrade. Then you add blood. Now, since cool yung temperature, it would not cause lysis, maintaining the red color of the RBC. But if it's cup naman, so you would use nutrient agar. Even though it's still hot, mainit pa rin, you add blood. And since the media is hot pa class, it would cause the RBC to lyse, turning the nutrient agar appearing as brown or chocolate in color. Now, when your RBCs are lyse class, it releases two factors, the X and V factor. The X factor stands for heme, while the V factor stands for NAD. Now, kaya siya considered na media used for fastidious bacteria is because itong X and V factor na to are required for certain bacteria such as yung Haemophilus and yung Neisseria. So your cap would appear brown color due to the lyse red cell, supports pathogens that require intracellular nutrients, especially critical for respiratory and genital pathogens. So this is the appearance of your cap. The fourth one is yung CNA nyo, or the Columbia Colistin Nalidixic Acid Agar with blood. So this is a selective and differential media used to isolate gram-positive bacteria. Composition, nutritionally rich, base with 3 peptone sources and 5% sheep blood. It contains antibiotic colistin, which would disrupt gram-negative cell membrane, and nalidixic acid, which inhibits DNA replication in gram-negative. So itong CNA nyo class is designed for isolating gram-positive. It kills or it prevents the growth of gram-negative bacteria through the help of these two antibiotics. So si colistin nyo, sinisira yung cell membrane ng gram-neg. Si nalidixic acid, it prevents DNA replication. So it supports differentiation of hemolytic reactions among gram-positive, prevents overgrowth by gram-negative organism in mixed culture. So this is your CNA. Next is eosin methylene blue or your EMB agar. So selective and differential din siya, but this time for gram-negative. It contains eosin Y and methylene blue dyes, which inhibits gram-positive and differentiates lactose fermenter. So, pang ano rin siya class, para siyang makongki. Nagdi-differentiate din siya ng lactose fermenter. So, if your bacteria is a lactose fermenter, tapos in-strict mo siya sa EMB, the lactose fermenter would appear as dark purple to black colonies. Strong fermenters like E. coli shows a green metallic sheen. So, pakitandaan class, if E. coli is grown in EMB, it produces a green metallic sheen. While non-fermenters such as your Shigella would produce colorless or translucent colonies. Relies on the fermentation of lactose, useful for enteric bacteria differentiation. So this is the appearance of your E. coli. It produces a green metallic sheen on your EMB agar. Next is the gram-negative broth. So this is an enrichment broth for gastrointestinal pathogens such as your Salmonella and Shigella. It contains sodium citrate, sodium deoxycholate, inhibits gram-positive organism, contains mannitol for early multiplication of gram-negative pathogen. Important note is that it needs subculture after 6 to 8 hours to prevent overgrowth by commensal. Enhances recovery of pathogens in low number. So ito yung gram-negative broth niya. Next is yung HEA or yung Hectowin enteric agar. This is a selective and differential media for isolating enteric pathogen Salmonella Shigella from stool sample. It contains bile salts and dyes to inhibit gram-positive organism. Carbohydrates, lactose, sucrose, and salicin test fermentation ability. Ferric ammonium citrate and sodium thiosulfate would detect hydrogen. They would detect hydrogen sulfide. So if it's lactose or sucrose fermenter sa HEA, they would appear yellow to salmon red colonies. Kapag non-fermenter like your Shigella and Salmonella, they would appear green to blue-green colonies. While Salmonella would produce hydrogen sulfide producing black-centered colonies. So pakitandaan klasa kapag hydrogen sulfide, ang color associated with that is black. So ito yung HEA nyo. So if you take a look at this class, yung may mga growth dyan na black yung center, these are usually your Salmonella. It produces hydrogen sulfide. Now let's go to the 8th one, yung McConkey nyo. So this is a primary selective and differential agar for gram-negative bacilli. It contains crystal violet and bile salts to inhibit gram-positive organism. Neutral red is the pH indicator. If lactose fermenter, pink to red colonies. Pag non-lactose, colorless colonies. Distinguishes enteric based on lactose fermentation. So isa siya sa mga widely used diagnostic bacteria. So this is the appearance of your MAC. So again, kapag lactose fermenting yung green nag-grow dyan, it will produce pink colonies. Pero kapag non-lactose, usually colorless. Next is your P agar or your phenyl ethyl alcohol agar. Selective medium for gram-positive cocci in specimens with mixed flora. Essentially, sheep blood agar supplemented with P. Functions: inhibits gram-negative rods, supports gram-positive cocci such as your strep, enterococcus, and staphylococcus. Cannot be used to interpret hemolysis pattern because alcohol interferes with red cell reaction. Then we have your modified Thayer-Martin agar or the MTM agar. This is an enrichment and selective medium for Neisseria gonorrhea and Neisseria meningitidis. Composed of chocolate agar base enriched with peptones, amino acids, vitamins, glucose, and antibiotics. So the antibiotics included are the following: colistin for inhibiting gram-negative, vancomycin for inhibiting gram-positive, nystatin for fungi, and trimethoprim to prevent proteus. Now, si Thayer-Martin yung class has a modification. The modification is known as the Martin-Lewis agar. Sa Martin-Lewis agar, instead of using nystatin, ang ginagamit mo na antibiotic here for fungi is ansamycin, which increase vancomycin concentration. Importance is that it ensures recovery of pathogenic Neisseria while suppressing normal flora. So ito yung MTM nyo. Para lang siyang chocolate agar class. Another one is the thioglycolate broth. So this is the most frequently used backup broth or semi-solid media. usually for identifying different oxygen requirements. So it consists of caseins, VIF extract, supplements, oxidation reduction indicator, resazurin, dextrose, vitamin K, and hemine. Agar 0.075% reduces convection currents, helping maintain oxygen gradient. It also contains sodium triglycolate, which is a reducing agent, lowers oxygen levels to create an anaerobic environment. Key concepts which would be include differentiates organism by oxygen requirement. So kapag near the surface, strict aerobe. Kapag diffuse growth, facultative. Pag restricted to the bottom, obligated anaerobe. Microaerophiles grow just below the surface, and gram-positive cocci would often produce clump growth. Now, si Resazurin class acts as an indicator. It turns pink in the presence of oxygen. Not classified as a differential medium in clinical labs. So, ito yung thioglycolate broth nyo, class. Next, we have yung XLD nyo. So, XLD is a selective and differential medium primarily used for isolating and differentiating Shigella and Salmonella species. It contains sodium deoxycholate, which would inhibit gram-positive organism and many gram-negative bacilli that are not enteric. It contains phenol red, which is a pH indicator, detects fermentation of carbohydrates, lactose, xylose, and sucrose. Now, bacterial appearance, if it's Shigella, it does not ferment lactose, sucrose, or xylose, meaning the colonies would appear colorless. While your Salmonella would ferment xylose initially, but the carboxylate lysine reverts to an alkaline reaction, making the colonies colorless. But as I mentioned, class, pag si Salmonella, it always produces hydrogen sulfide. Therefore, kahit originally colorless siya, but since there is hydrogen sulfide, it would now produce a black color colonies. Then, non-pathogenic coliforms in XLD would ferment one or more carbohydrates, turning the colonies into yellow. Designed to highlight Shigella and Salmonella from stool culture. Differentiates enteric pathogen based on carbohydrate fermentation, lysine decarboxylation, and hydrogen sulfide production. So, ito si XLD nyo. So, on the left one is the Shigella. So, colorless. But Salmonella is originally colorless as well. But dahil nga nagpo-produce siya ng hydrogen sulfide, it now has black center, making the colony appear black. Now, how would you prepare artificial media, class? So you need to make sure that you sterilize this to eliminate contaminant. And the most common method of sterilization is autoclaving. So prepared at 121 degrees centigrade for at least 15 minutes. After sterilization, molten agar is cooled to 50 degrees before pouring into plates or adding heat-sensitive supplement. Heat labile substances such as your serum, antibiotics, and vitamins cannot withstand autoclaving. So kapag yung media nyo, class, would contain serum, antibiotics, vitamins, hindi ka pwedeng gumamit ng autoclave. You're not allowed to use autoclave for sterilization. Instead, you would use filtration and lower temperature methods. For filtration, you would use 0.2 to 0.45 micrometer filters. For lower temp method, 116 to 118 degree centigrade. Now, there's what we call cell culture. Now, there are certain bacteria class that are unable to grow in artificial media. They require live organism to grow, and these are known as obligate intracellular parasite. When you say obligate, they are required. Intracellular meaning inside the cell. So basically, these are bacteria that are required to live inside a cell or a host. An example of this is your chlamydia, rickettsia, and rickettsia-like organism. Now, cell culture would involve layers of viable host cell grown in flasks, tubes, or on surface where bacteria can infect them. Now, detection of growth can be done by observing changes in cell morphology using nucleic acid detection or antibody-based stain to reveal bacteria inside the cell. Toxins can also be detected in cell culture such as Clostridioides difficile cytotoxin, viruses, which are all obligate intracellular parasite, also requires this method. Now let's talk about chlamydia. Now they would require living host cell. Common culture method. Now they would grow on cell culture line, McCoy cells, HeLa cells, or other tissue culture. It would also grow on embryonated chicken egg, specifically on the yolk sac. Then we have rickettsia. So rickettsia is also an obligate intracellular, does not grow on routine media. Cultured in embryonated chicken egg, animal tissue, such as the mice, guinea pig, peritoneal cavity, and brain inoculation. Cell lines, it grows on cell lines, verocells, human endothelial cells. Then rickettsia-like organisms would include your Coxiella burnetii, Ehrlichia, Anaplasma, and Mycobacterium leprae. So si Mycobacterium leprae nyo class is known to cause your leprosy. Now, culture method for Coxiella class, yolk sac of embryonated eggs or cell culture. For Ehrlichia and Anaplasma, it uses leukocyte culture and thick cell lines. For Mycobacterium leprae, it would grow on mouse foot pads. So, sa paan ng mga mice class and armadillos, wherein it is the natural host used to multiply bacilli in bulk. Now, environmental requirements for your bacteria are oxygen and carbon dioxide. So, na-explain ko naman yung oxygen, but let's have a recap. If they require oxygen, they are known as obligate aerobes. Cannot grow in oxygen, obligate anaerobe. Facultative anaerobe may grow with or without oxygen. Microaerophiles grow best at reduced oxygen, 20% or less. Then aerotolerant survives without oxygen but do not use it. Now, yung certain bacteria nyo class would require high levels or elevated level of carbon dioxide. Usually 5 to 10%. So pag ganun ang bacteria nyo, they require 5 to 10% carbon dioxide. They are known as what we call caphnophilic bacteria. Now, temperature class, most bacteria grow at 35 to 37 degrees centigrade. But there are certain bacteria, such as your 30 degrees centigrade for fungi and other bacteria, 42 degrees centigrade for Campylobacter jejuni, and 4 to 42 degrees Listeria monocytogenes and Yersinia enterocolitica. But they prefer 20 to 40. Itong dalawang to class, these two bacteria, yung Listeria nyo saka si Yersinia, both of them are known to cause food poisoning. Let's say hindi ka mahilig, you're the type of individual that does not clean your refrigerator. Siguro naglilinis ka lang ng rep mo once a year. There's a possibility that your Listeria and Yersinia would grow in the refrigerator. And when that happens, and you store food in the rep, Listeria and Yersinia would infect the food, and in some cases, it would cause food poisoning. Then there's also your pH requirement. So most bacteria would require a neutral pH 6.5 to 7.5. Then moisture. So water is critical component for broth and agar. During evaporation, incubation, evaporation which may occur, which would cause or stress or lyse the bacterial cell. So to maintain adequate humidity, seal the agar plate and use humidified incubators. Now, methods for providing optimal condition. So incubate at 35 to 37 degree. Some media would contain pH indicators and cannot be used in carbon dioxide incubator because excess CO2 lowers pH. So if that's the case, use alternative class such as carbon dioxide generating system. Yung mga gas pack yung nagpo-provide ng sachet, provide controlled CO2 for fastidious organism such as your Neisseria. Incubation duration varies, so mostly routine bacteria 24 to 48 hours, 1 to 2 days. Anaerobes may take weeks, while Mycobacteria often requires extended incubation. For Mycobacterium class, usually 8 to 10 weeks. So here are the references. So that ends activity 5 media preparation. Thank you.